ಧನ್ಯವಾದಗಳು. ಪ್ರಥಮವಾಗಿ ತಮಗೆಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ ಮತ್ತು ಶುಭ ಅಪರಾಹ್ನ. ಮತ್ತೆ ಮಾತಾಡ್ತೇನೆ. ನಿನ್ನೆ ಬೆಳಿಗ್ಗೆಯಲ್ಲಿ ಒಳಗಡೆ ನಮ್ಮ ಕರ್ನಾಟಕ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಅಕಾಡೆಮಿಯ ಪ್ರಾಂಗಣ ಒಳಗಡೆ ಬರ್ತಾ ಇದ್ದ ಸಂದರ್ಭದಲ್ಲಿ ಡಾಕ್ಟರ್ ಅಯ್ಯಪ್ಪನ್ ಸರ್ದು ತುಂಬ ನೆನಪಾಯಿತು. ಯಾಕಂದರೆ ಇದೇ ವೇದಿಕೆಯಲ್ಲಿ ಸರ್ ಒಂದು 2-3 ವರ್ಷಗಳ ಹಿಂದೆ ಒಂದು 3-4 ಕಾರ್ಯಕ್ರಮಗಳನ್ನು ಅವ್ರ ಜೊತೆ ಮಾಡಿರುವಂತಹದ್ದು. ಇವತ್ತು ಅವ್ರು ನಮ್ಮ ಜೊತೆ ಇಲ್ಲದೇ ಇದ್ದಿರಬಹುದು, ಆದರೆ ಅವ್ರದ್ದೊಂದು ಸರಳತೆ ಇದೆ ನೋಡಿ, ಸಿಂಪ್ಲಿಸಿಟಿ. ನಿನ್ನೆ ಕೂಡ ತುಂಬ ವಾಗ್ಮಿಗಳೆಲ್ಲ ಅವ್ರ ಬಗ್ಗೆ ಎಲ್ಲ ತುಂಬ ಎಲ್ಲ ವಿಚಾರ ಎಲ್ಲ ತಿಳಿಸಿದ್ದಾರೆ. ಅವರ ವಿಜ್ಞಾನದ ಬಗ್ಗೆ ಜ್ಞಾನ, ಅವರ ಲೆವೆಲ್ ಆಫ್ ಕಮಿಟ್ಮೆಂಟ್, ಮತ್ತು ವಿಶೇಷವಾಗಿ ಯುವಕರಿಗೆಲ್ಲ ಒಂಥರ ಎನ್ಕರೇಜ್ಮೆಂಟ್ ಮಾಡುವಂಥದ್ದು, ಇದೆಲ್ಲ ಅವರೆಲ್ಲ ಸದ್ಗುಣಗಳಂತ ನಾನೇನು ಮತ್ತೆ ಹೇಳುವಂಥ ಒಂದು ಅವಶ್ಯಕತೆ ಏನಿಲ್ಲ. ಅಟ್ ದ ಮೋಸ್ಟ್ ಅವರೆಲ್ಲ ನಡೆದು ಬಂದಿರುವಂಥ ದಾರಿಯಲ್ಲಿ ನಾವೆಲ್ಲ ಯುವಕರೆಲ್ಲ ಮುಂದೆ ಹೋಗೋಣ ಅಂತ ಆಶಯದೊಂದಿಗೆ, ಮತ್ತು ವಿಶೇಷವಾಗಿ ಐ ರಿಮೈನ್ ಹೈಲಿ ಥ್ಯಾಂಕ್ಫುಲ್ ಟು ಯು ಸರ್, Dr. Idya Karnasakar, who was primarily responsible for linking me with this particular day's event. And I remain equally thankful to the Chief Executive Officer of KSTA, Dr. Ramesh and Mr. Srinivas, and the team of KSTA. Yeah. I need not have to emphasize much as far as the global threat, as far as the AMR issue is actually concerned. It is not just the threat at the global level, but also at the national level, regional level, as well as at the local level. And what is the actual impact of antimicrobial resistance in India? As far as the data which is already generated in the year 2019, nearly 3 lakh people have actually died of this AMR. And what is the projected figure? By about 2050, it is expected that maybe about 2 million, 20 lakh people could be succumbing to this particular situation. But nevertheless, India is not sleeping. India is also extensively responding to this particular AMR situation, and then trying to evolve several strategies how to contain this particular menace. And as far as my presentation is concerned, I am not going to talk much about the technical aspects of it, and because of 2 important reasons. Because I don't want to stand between you and the lunch. Then second reason is I need to leave out of station by today evening itself. So let me quickly rush through the presentation and then take it forward. We know the government of India has already launched this national health policy way back in the 2017, and our Minister of Health and Family Welfare, they are also very closely collaborating with the WHO, and tackling of AMR is considered to be one of the top 10 priorities. And then in 2014, separate schedule H1 was actually incorporated, wherein they wanted to regulate the sale of antimicrobials in the country just across the counters. That is what was actually put forth very effectively by the first speaker of yesterday's session. FSSI, they have also banned the usage of antibiotics in the fisheries. We have the fisheries experts here. And then they have also instituted this maximum capping levels of drugs usage as far as the growth promotion in case of the food animals is concerned. What are the various national initiatives taken to prevent this AMR situation? AMR programs, AMR containment, surveillance and research is also emphasized. International collaborations, because just now the previous speaker, he mentioned how important it is to get connected with the intersectoral coordination, and it is important that we need to work under the One Health umbrella and then get connected with the like-minded people. And then antibiotics stewardship was also emphasized, and then control of overuse or misuse of the antimicrobials or antibiotics. This is the National Action Plan for Antimicrobial NAP AMR. This was already launched in India in the 2017, and there are certain priorities which have already been discussed. Yeah, then coming to the states, as far as the document 21 that was actually started in 2018, Kerala has been the first state in the country to start this SAP State Action Plan. Subsequently, even Madhya Pradesh, Delhi, even Karnataka is also in the pipeline. This is also initiated already in the country. I think as of now, at least 6 states must have submitted the proposal to Government of India for institution of this SAP care. Why there is an urgent need to explore the innovative alternatives to antibiotics? We know very recently, just in the month of November 2024 itself, naphthramycin was actually that was discovered after 30 years of the struggle. And then at present, I believe there are only about some 27 drugs which are in the pipeline that needs to be taken forward as far as their targeting of WHO priority pathogens is concerned. So discovery of new antibiotics technically demands is technically demanding, and financially it is not that feasible. So because of these reasons, globally as well as nationally, we are actually thinking of exploring the alternatives to these antibiotics so that we will be able to tackle this AMR situation effectively. Major mechanisms of action of alternatives, I don't think I need to get into the technical aspects of these various mechanisms based on which these alternatives to antibiotics could be functioning. There are four major categories of alternative strategies. One is to target the antimicrobial resistant enzymes, then targeting the antimicrobial resistant bacteria, drug delivery systems as Madam Indrani Karuna Sagwa just now she was mentioning, and then physicochemical methods are also being explored. Yeah, these are some different kinds of alternatives, right from vaccines. My friend Dr. Hegde is going to talk to us extensively after this presentation as far as the major role of using these vaccines effectively, especially in the veterinary sector. Because let us undertake vaccination in the livestock in the larger scale so that the disease itself is very effectively prevented. So there is no need of using the antibiotics on these diseased animals. Then immunotherapeutics, and then CRISPR-Cas, this is one of the effective molecular tool which is being employed in most of the laboratories. Then probiotics, microbial therapies, phage therapies. Just now Madam actually elaborated extensively on the usage of bacteriophages and different versions, and then molecular changes to be made in these phages. Then fecal transplant is being done in case of animals. So I am going to talk to you in brief about this fecal transplant mechanism. Then nanobiotics and enzybiotics are also employed. Then stem cell based antimicrobial peptides. Now coming to probiotics, prebiotics, post as well as the symbiotics. They basically interfere with the pathogen attachment to and entry into the gut mucosa. They produce several antitoxins. They produce several antibacterial substances such as bacteriocins as well as the organic acids. They help us to destroy these toxins and restoration of the gut environment. This is very, very important aspect. Then they also bring about the immunomodulation by triggering the protective cytokine production. This is just the pictorial expression of what I just said in the previous slide. Coming to the phage therapy, need not have to again get into the details. Yeah, Nitya University has been one of the pioneer institutions in the country itself, and thanks to Dr. Edi Karuna Sagar sir for having taken initiatives to establish this pioneer FAO Center for Antimicrobial Resistance. In fact, a couple of weeks ago, one of our partners from Royal Holloway University of London, we had a chance to interact with Professor Edi Karuna Sagar, and let us see how best we can take it forward. And one of the priorities of this FAO Center is to develop these alternatives to antibiotics as well. Yeah, this is again the data which they have already generated. Madam, in fact, I got it from Dr. Akhila. So they have already got a plethora of bacteriophages, and then so they are working a lot on development of phages and then phage therapy, how to make it more safe, and then targeted alternatives to combat antibiotic resistance. This is what little we have done in association with the Nitya University a few years ago itself. Yeah, this is the kind of the collaboration between us and the NUCSER, wherein we procure the bacteriophages. It is like win-win situation. They share the bacteriophages, we share the isolates to them, and then one of our PhD students in our KVFS also, she also as a part of our PhD program, we try to look for the lytic activity of these bacteriophages that we have procured from them on different isolates, Staph aureus as well as E. coli, and then we found them effective. This is what is already initiated and completed by Dr. Nagendra Reddy. He is going to be the next speaker. In fact, we had a collaboration which is funded by the Department of Biotechnology, that was primarily on boil mastitis, and therein also we have done a lot of capsular typing and then development of some vaccines, and then some phage related work was also initiated. And this is what he has already done with his team at NIB, which is again funded by the Department of Biotechnology. They have also tried this E. coli, enterococcus and Staph aureus to look for the lytic activities of these phages. Yeah. Now coming to the commercial aspect, this is one thing that I just wanted to surf through. What are all the different organizations or agencies or universities or institutions in India who have been associated with one or the other way of the activities pertaining to these alternatives of antibiotics? This is one. Aristogen Biosciences is one of the pioneer company in Bangalore itself, and they have developed several commercial products which are comprising of these bacteriophages. These are some of the commercial products that they have already released. They are commercially available for control of these infectious diseases in case of poultry as well as in aquaculture. VibZ for controlling these Vibrio infections in shrimp, and then Vibrio Shield again for control of fibrosis in aquaculture. Holy Shield for control of this pathogenic E. coli, and then Sal Shield for control of pathogenic Salmonella in case of poultry. These are all already released in the market. Then there is another company called Gangajan, who are also deeply through the development of these bacteriophages and clebicins, and then actolysine P128. This P128 appears to be highly effective against the Staph aureus, and we propose to collaborate with them. And then we have hundreds of these Staph aureus isolated from the Boyan mastitis cases, and we would like to check the activity of these P128 product on these MRSAs. Yeah, here we have actually plethora of the agency names, products, bacteria which are already published. Then the next important alternative is Ayurveda. You know how much government of India is actually supporting through the Ministry of Ayush, primarily for the purpose of reviving the ancient system of medicine. In fact, we Indians, we have been practicing Ayurveda since last several hundreds of years, and this is being pushed through the government of India through the Ministry of Ayush. Yeah, Ashwagandha, curcumin. We have been using them since ages. It is nothing new to us. Nowadays, the thinking is initiated how to generate the data by getting the extract of these Ayurvedic products, and there are these basil, clove, lemon balm, then pomegranate, benzoic acid, cinnamic acid, and eugenol. They are all present already in the herbs which are routinely being used, and they are found to be effective against not just the gram-positive as well as gram-negative bacteria, but also against some multi-drug resistant pathogens. Here again the biomedical applications of plethora of herbal materials, whether it is cancer therapy, cardiac involvement of neural tissues, or even skin regeneration. This is again how beneficial these curcumin derived extracts are. Again, we can see just the seeing is itself is believing, need not know to explain, including the tomato, grape, soya bean, and then green tea. Green tea is offered in almost all the offices wherever we go. So this is becoming very, very popular with respect to its antimicrobial activity. This is about ginger. Most of us, we enjoy having the ginger tea. This is one of the very, very important the development. In fact, they have extracted they extract the active ingredient from ginger, and it is also very well characterized, and these are all the various activities of the extract obtained from ginger. Yeah, now we have the contributions made by one of the very well known popular institutions in Bangalore, Transdisciplinary University. We have the former director Dr. Verma, who is the pioneer in this field. We had also chance to be having some kind of collaboration. They are also deeply into this holistic health based approach, Transdisciplinary University. Then there is another institution which has come up very recently, perhaps some 5 or 6 years ago, that is the School of Ancient Wisdom in Bangalore. This institution is actually based in the northern part of Bangalore, and they are deeply involved in this capacity building, training, local resource development, curriculum development. That's what I was just discussing with Dr. Ghaffar. And then knowledge dissemination through the Ministry of Ayush, then research and publication, government recognition. As a part of this government collaboration of this particular institution, they have already developed these 43 Ayurvedic formulations. This is one very important contributions of this particular institution. Then herbal formulation from ICR, Indian Council of Agriculture Research. IVRA is the institution affiliated to ICR, Indian Veterinary Research Institution. They are also very much into development of so many alternatives to these antibiotics. They have got some antitic phyto formulations. They have got some IVRA herbo mineral acaricide formulation. You see, just like microbes, even acaricides are also triggering the development of anti-acaricide, this kind of resistance by these ticks. And in fact, one of the audience, when Dr. Veena, she did a very good work as far as enzymatic resistance is also concerned from parasitology department. These are some more products released by ICR IVRI. This is about one more institution from Tamil Nadu. They have got these various leaf extracts of Ocimum americanum, and the leaf extract was found to be having very good antibacterial activity for Bacillus, Clostridium Klebsiella and Candida and some and a couple of other fungal species. This is about the antibacterial activity of Phyllanthus MRS. And here one thing I just wanted to emphasize, you see this is actually the standard, and the aqueous this extract obtained from this particular Phyllanthus MRS plant, it was working almost on par with the standard but not the acetone extract, and this was demonstrated to be having antibacterial activity against various species of bacteria including both gram positive as well as gram negative. This work is actually done by one of my colleagues from KVFS itself, Dr. N.B. Sridhar. He is in fact a professor and head of a pharmacology department at Shimoga Veterinary College. He is into very much into this ethno-veterinary medicinal products. He uses them and he actually tries to look at the efficacy of these ethno-veterinary medicine, especially for treating the mastitis cases in case of the villages. Here he is actually using some of the extracts of garlic and then capsicum, then sesame and then gallium apare plants. We know several commercial companies are also playing major role. We know about these Indian herbs. This is one of the pioneer institution. It is there since last more than 70 years, and most of their products are actually based on the herbal products itself for improving the animal health. Then two more organizations are natural remedies. They are very much into this research on advancing the phytoomics effects as far as the global food safety is concerned. Then Himalaya Drug Company. Earlier this company was actually known as the Himalaya Drug Company. Now it is actually renamed as Himalaya Wellness Company. And they are also one of the popular organizations across the globe who are into so many herbal products and for the purpose of both human health as well as the animal health, livestock health. This is about homeopathy. Homeopathy is extensively practiced in case of human population. But some of our colleagues in the university itself, they are also deeply through this homeopathy practice in case of animals. And most commonly, at least some of us are very familiar with this tincture calendula and then belladonna. In addition to these, there are several other homeopathy products which are being extensively practiced for treating the skin infections, the cuts, then to treat the open wounds, then insect bites and including the bruises. Commercial products are already available and this is being practiced by some of the field veterinarians. Then coming to yet another alternative, that is antimicrobial peptides, and some of the antimicrobial peptides have been found to be very effective against MDR pathogens as well. Stem cell derived AMPs are also reported, that is human mesenchymal stem cell synthesized factors similar to AMPs, and some of these AMPs derived from these MSCs were reported to be having antibacterial activity with respect to MRSA, methicillin resistant Staphylococcus aureus. Still a lot more research needs to be done. And then combat implants associated infection. See, this is how the concept of biofilm actually arrived way back in 1970s. Because of the implants, the biofilm formation was triggered and it was really tough to treat this biofilm based bacterial community. And these AMPs derived from MSCs were found to be having very good activity, antibacterial activity against E. coli and then Staph aureus as well as Staphylococcus epidermidis. Yeah, one more contribution by IVRI. They have developed these chirosong encapsulated AMPs, antimicrobial peptides, and were found to be having very good activity against MDR salmonella enteritidis. This is one of the promising products of ICR IVRI. And then with respect to maintaining the intestinal integrity in case of the suckling piglets, the young piglets, they have found out that the bovine lactoferrin as well as the pediococcus FT28 probiotic interventions have really helped them to improve the integrity of the suckling, the intestinal health of the suckling piglets. This is about immunomodulators, and we have again good number of immunomodulators which are really in combination with antibiotics or other products could be of some benefit to us to tackle these antimicrobial resistance situations, cytokines, interleukins. There are ways, say some of these probiotics, they also trigger the production of these cytokines, which is of direct benefit to the clinicians. Then immunotherapeutics and vaccines. As I already told in the beginning itself, extensive vaccination is actually practiced in livestock or even poultry, and we have Dr. Ratnama, who is actually the director of this premier institution of our Karnataka Veterinary Animal Fisheries Institution. Her institutions make very good number of these vaccines to control these viral diseases as well as bacterial diseases in case of animals or in case of birds. In fact, she was my colleague and we were together, and since last 1 year she is heading this very prestigious institution, and I told you how important it is to prevent the disease. Prevention is always better than cure. So if you can have large scale vaccination to control these diseases, the occurrence of disease doesn't arise at all, whether it is anthrax, whether it is BQ or HS or whatever it is. So in that case, in that context, extensive large scale vaccination is definitely one of the best alternatives to tackle this AMR. And Dr. Hegde, he is a pioneer, he is a immunologist, and he is going to talk in detail about the role of vaccines to tackle this situation. Again, pertaining to the recombinant vaccine technology, I am sure Dr. Hegde is going to take it forward. I was talking to you about this fecal microbiota transplant. As I understand, even in rural areas, they have been practicing it since ages. What exactly they do? To stabilize the integrity of the intestinal mucous membrane. Supposing there is some disturbance in the intestinal mucous membrane. For example, this is actually very well worked out. Clostridium difficile, that results in some intestinal disturbances. As you can see in this illustration, we introduce these helpful microbes, which is actually collected from the fecal matter from the healthy cattle, then inoculated orally, and then it will actually destabilize these pathogenic bacteria, and then this will be establishing there in the mucous membrane, in the mucus of intestinal tract, and successfully this was able to manage the conditions such as IBD, inflammatory bowel disease, as well as irritable bowel syndrome. This is also common even among the human beings. Then this was found to be very clearly colonizing the MDR pathogens, and most importantly, this is very cost effective approach, and this can be done very fast. So these are the two major advantages of this FMT approach. Then CRISPR-Cas, this is one of the most commonly talked molecular tool, and this can directly go and attack the sequence which is responsible for expression of factors associated with AMR, and this is also being practiced. And in this particular publication, it says that this CRISPR-Cas complex was able to target the MEK gene. We know how important this MEK gene is as far as conferring AMR is concerned. This is about nano antibiotics. Much talked of. Both metal as well as metal oxide based nano antibiotics have actually become popular of late. And among the all the classes of these nanoparticles, silver nanoparticles has been extensively used, and it was found to destroy these AMR pathogens by various other mechanisms which I have already mentioned. Whereas as far as candida infections are concerned, MDR candida infections are concerned, bismuth nanoparticles are found to be really beneficial. And then there are some of the preparations which are already available for topical application based on these nano antibiotics. Yeah, in fact, we also wanted to try with the antibacterial activity with special reference to MRSA again in our own lab by using only the nanoparticles which I have already mentioned there, like silver oxide, zirconium oxide, hydroxyapatite, and aluminum oxide. But to our surprise, none of these nanoparticles on their own were able to kill the bacteria, which are actually MDR, and maybe we need to go for conjugation of these nanos with some other particles and then give a try. And this is just observation made by one of our students. And then yet another PhD student in the university itself, she in fact tried to ascertain the role of nanothallium and nanocurcumin, and this was actually found to really impart the natural immunity, and thus very well supported the growth of the broiler chicken, as you can see there. Then mesenchymal stem cell condition media based biomesh. This is another finding. So what they have done, the MSCs, this conditioned media was actually mixed with the laminin gel, and then this leads to formation of biomesh, and this was applied on the wound that was actually created on the laboratory animals, and this is found to be a very good scaffolding augmented with the mesenchymal stem cell condition. This is one, but this is yet to be popularized, and this is just in the creation of this biomesh is just one of the recent findings. Then AMR declaration test, Chennai declaration. Just now the previous speaker himself is actually the brain of this particular, and I don't think I need to mention much about this Chennai declaration, wherein they were collaborating with various governmental as well as non-governmental organizations to tackle this issue. Down to earth, this is one food service company that was actually working with the poultry farms primarily for the purpose of elimination of antibiotic use by focusing on the bird health as well as by employing the proper farm management. DBT, BIRAC, they are also funding a lot for the AMR based research activities, and I'm sure, I think Dr. Agde is also funded under this or a BRIC for whatever the work they have already initiated. Yeah, it's a BRIC, I believe. Then development of alternatives to antibiotics and development of new antibiotics. These are the two important mandates under this DBT BIRAC program. Yeah, this is one network SAMU that was actually constituted hardly some 4 or 5 years ago. That stands for Network of Veterinary Education for Sustainable Antimicrobial Use and Tackling of AMR. This is actually acronymed as network SAMU. And just now I was actually talking to someone about the role being played by our collaborator. In fact, we were funded by DBT for one particular program called Drivers of Antimicrobial Resistance in Poultry. Dr. Agde himself was the chief investigator for India and one of the partners from University of London. So he was collaborating, and recently he was also in India, and we had a wonderful interactive session with the FAO Center with reference to Dr. Idekarna Sagar sir, and let us see sir how best we can take this collaboration further. Then recently Government of India had also approached various veterinary institutions as well as medical institutions in India just to check what are all the kinds of antibiotics in their respective regions that are being used, whether we need to have so many antibiotics or whether we need to have really so many antimicrobials. But we cannot think of getting away completely from the antibiotics. If not for growth promotion of a poultry or any other species of animals, at least to treat the emergency conditions, we have to have these antibiotics. But at the same time, several organizations, several institutions and universities are into conducting the research as far as studying the efficacy of alternatives to antibiotics are concerned, and what we have given our opinion to the Government of India, Department of Animal Husbandry and Dairying, that we have to have a sort of One Health based collaborative approaches, intersectoral coordination. Then we have to have some kind of a mechanism wherein the therapeutics should be used only in case of food animals, not the alternatives, not the antibiotics or antimicrobials, just as a growth promoter. And we will have to emphasize, take up lot of research on all these alternatives to antibiotics, generate the data, try to identify the active ingredients in these alternatives, and then we should be in a very confident position to say that yes, this works very well. Although these alternatives may not be working so effectively independently, but at least in combination, so that at least the antibiotics or antimicrobials dose could be minimized, and then this kind of research will have to be carried out, and then let us hope for the best. And to conclude, alternatives to antibiotics have a bright future, no doubt. There are numerous hurdles. ಈಗ ಹೇಳ್ತೀವಲ್ಲ, ಒಳ್ಳೆ ಕೆಲಸ ಮಾಡಬೇಕಿದ್ರೆ ಏನಾದರೂ ಒಂದು ಪ್ರಾಬ್ಲಮ್ಸ್ ಇದ್ದೇ ಇರುತ್ತೆ. ಈಗ ಮಲ್ಕೊಂಡವ್ರು ಯಾವಾಗೂ ಅಡುಗೆ ಬೀಳೋದಿರತ್ತಾ, ಓಡ್ತಾ ಇರೋನೇ ಅಡುಗೆ ಬೀಳೋದು ಯಾವಾಗ. ಸೊ ಲೆಟ್ ಅಸ್ ಕೀಪ್ ಟ್ರೈಂಗ್ and let us get connected with the like-minded people under the One Health umbrella and try to understand and explore various strategies to beat these AMR pathogens. And extensive research is the need of the hour as far as studying the efficacy of alternatives to antibiotics is concerned, so that we can tell the world that yes, these are the alternatives that we have in India, and this can definitely be working in complementation with the antibiotics or in combination with the antibiotics. So thank you so much. Let's fight this antibiotic resistance.